the next topic by Mr. T.Seipelt, Abbott Nutrition, USA. Mr. T.Seipelt is the Director of Product Stewardship and Food Safety at Abbott Nutrition, a division of Abbott Laboratories. He has worked in food safety and chemical contaminant analysis for over 30 years. In his current role, he is responsible for the development of global food safety programs, ingredient and packaging chemical risk assessment, and management of the emerging issue program. He holds a Bachelor of Science in Forensic Chemistry from Ohio University. So here I would invite Dr. Tom Shiplet to speak on importance of monitoring melamine and cyanuric acid in milk and milk products. So can we please have the AB?
Well, I'd like to start with saying thank you for the opportunity to present. I'm sorry that it has to be virtual and I'm not able to be there in person. I was looking at the calendar, and in November last year, I was actually in India working with laboratories in Mumbai and Bangalore. And then in November this year, I'm not able to travel. So 2020 is a very different year, and I'm sorry I'm not able to join personally. I do appreciate the opportunity to talk about food authenticity and food safety. And just a brief introduction to myself. My name is T.Seipelt. I work for Abbott Nutrition. We're based in Columbus, Ohio in the US. I've been with Abbott for about 35 years now and been working in food safety and chemical contaminant testing most of that time. When we talk about food authenticity, we can also talk about food adulteration. Food authenticity is the positive aspect. We want to verify the identity, the integrity of the food, make sure that it is what we want, that it has the right nutritional content, that everything is good. Food adulteration is the other side of that coin. We're really trying to do enforcement type activities. We're looking for things that aren't supposed to be there, things that are not the right quality, things that, in the case of adulteration, they may have been added intentionally. It's different from a contamination event where something might get into the food by accident. Adulteration, people are typically adding things or changing the food intentionally. Often that's modified by economics. They might dilute the food, they might add something else to it to try to increase its apparent value. Could be a color, could be a different type of flavor. But they're really doing it to try to fool the buyer and to make the buyer pay more than what the product is actually worth. Food adulteration is an ancient problem. It's been around for almost as long as we've had written history. It was present in ancient Rome and Greece. Documented cases of wine adulteration where it was either diluted or perhaps herbs or spices were added for flavoring. Sometimes even lead was added as a preservative, which then, of course, created other safety issues. So food adulteration is not new, but what has been new in the last 12 years, really since 2008, is a very acute awareness of the potential for these adulteration events to not just be economic, but to actually cause food safety implication. The melamine incident in China, we'll talk a lot about that and what drove that. But that really changed the way that we approach food safety and food adulteration, and the way that we test food and the way we try to monitor to assure that the food is the quality that we need, that it's the safe product that we need. When people are trying to do things for adulteration, you can never underestimate their creativity. As soon as we find a way to detect what they're doing, they're going to find another way. So we always have to be vigilant. We always have to be flexible to try to predict what they might do, to try to stay on top of what they might be doing. Melamine really hit the news in 2008. It was an previously unknown food safety and food adulteration issue. In 2008, the headlines really started to explode when melamine started to be detected in infant formula. It was in milk powder, which was then used to make infant formula. And in China, there were hundreds of thousands of infants who were made ill. Several actually died. So it became a real problem. As I said, it was previously unknown. So there was a lot of effort that went into trying to understand what was happening, how did this get into the food, how could we prevent it. So it was a very dynamic process for about 18 months. After that, we began to understand what the problem was, and it kind of went into a routine monitoring mode after that. We'll talk about why melamine was added. And I said that people who are committing food adulteration can be very creative, and they can be very smart. Melamine was added by some smart people who were trying to fool the system, but it continued to be used by people who really did not understand why it was being used. In the lower left here, you can see that melamine powder was actually added to animal feed so that it was fed to cows and got into the milk that way. It became known as protein powder. And we'll talk about why melamine is associated with protein. But because of that association, the farmer, who didn't really understand the science and the chemistry behind it, used it to enhance the milk. They didn't know why, but they knew that it could be there. What we've seen in the last 12 years is that this continues to be a practice that pops up every now and then. Despite all the issues with melamine, milk powder or protein powder is still known, and occasionally we'll get back into the supply chain. So we continue to have to monitor and be vigilant. So how did melamine come to be, and how did we find out? It really started several years before we found it in the human food chain and found it in infant formula. It was found first in pet food. There were multiple incidents where dogs and cats were dying after being fed the pet food. And it took several years to really figure out what was going on. In 2007, the detection of melamine was finally made, and the connection of melamine to the pet deaths was finally starting to be drawn. And as that was just beginning to be figured out, it appeared in China in infant formula. And so we found that it was being added intentionally to diluted raw milk, and it artificially then inflates the apparent protein value. And we'll talk about that in just a minute. What was happening, though, is that as melamine was being detected, and the people who were committing the adulteration added a little, and they got away with it, and so they added a little more, and they got away with it. And after a while, it got high enough that it started to cause food safety problems. People started getting sick. And there were about 300,000 Chinese infants and 6 fatalities in the first 6 or 7 months of this incident. It represented one of the largest deliberate adulteration events that was known at the time. And it really changed the way we address the food safety system. The impacts to the China milk industry continue today. FSSAI still has a ban on China milk products. And so the long-term implications were severe. The infant formula company, the milk company called Sanlu in China, that was associated with the event, is now out of business, and many of their executives were convicted of the crime. So why melamine? Why was it added to the milk? The chemical structures are shown here, and the most important thing to point out is the high amount of nitrogen. Over 60% by weight of melamine is nitrogen. It was added to artificially elevate the apparent protein value. The Kjeldahl method for protein analysis actually measures nitrogen. It's not a direct measurement of protein. So by adding nitrogen, a nitrogenous compound to the milk, it inflated the apparent content of the protein. By doing that, they were able to dilute the milk and sell it at a significant price increase. So it was a very profitable adulterant. Melamine's very common. It's used as a plastics monomer. It's used in some food coatings. So as regulations were being developed, we had to account for the allowed uses of melamine because traces might be present in the food. Same thing with cyanuric acid. It was related to the safety issues with melamine. It's a closely related compound. It can be a degradation product of melamine, but it's commonly used as a chlorine stabilizer. So when we're doing water treatment for food use or for drinking water, cyanuric acid can be present to stabilize the hypochlorite that provides the chlorine to do microbial control of the water. It's also a common chemical for swimming pools. So both melamine and cyanuric acid are very common, fairly readily available compounds. Because there are allowed uses in food contact or in food itself, either of these compounds might be present at trace levels in food. The real problem became when these were used as adulterants and the levels became much higher. So from a chemistry and toxicology standpoint, why did this become a food safety problem? Again, the structures for melamine and cyanuric acid are shown. The two compounds, as they get more concentrated, begin to form hydrogen bonds and they're insoluble. This aggregate, the melamine cyanurate aggregate, is insoluble. So if these are ingested and then as they're trying to be excreted through the kidney, they begin to concentrate in the kidney as water is removed to concentrate the urine. They form crystals, and actually in the kidney itself, you begin to get crystal formation and kidney stones. So this aggregate then caused renal failure, and that's why some of those children died, and that's why so many were sick. From a toxicological standpoint, melamine has been reviewed by JECFA, the European Food Safety Authority, US FDA, and TDIs for these compounds have been established ranging from 0.2 to 0.6 milligram per kilogram body weight. The important thing to note, though, is that there aren't data available to allow a calculation of a TDI when there's cyanuric acid present. The formation of these crystals really has not been studied enough to know what acceptable levels might be. And so in general, we take a zero tolerance approach. If melamine is present as an adulterant or even as an allowed part of the food use, we need to make sure that cyanuric acid is not present so that we're not causing unforeseen food safety problem here. Just to make sure that we understand the overall impact of the melamine issue in 2008, the range of concentrations that were found in various food types, it was not just related to milk or milk powder. It was not related to just infant formula. It showed up in multiple food types, things that contain other dairy ingredients, candies, baked goods, all kinds of different things. And the global nature of the supply chain really is shown on the right-hand side there, because it was really an issue that seemed to be isolated to China, where the adulteration events were taking place, but it was detected in food products all over the globe in the first year of the incident. From a regulatory standpoint, there are established standards. These standards are focused primarily on melamine itself. That's what we have the actual safety data for. It has established TDIs. And the primary point of the regulations was to be able to monitor and enforce the potential for melamine adulteration. Cyanuric acid is recognized in many of the supporting documents as a mitigating factor, but it's not included with a maximum level and not included in the regulation. When WHO, FAO looked at it for codex limits, these limits have been adopted by FDA in the US. It's also been adopted by FSSAI in India. 1 milligram per kilogram in an infant formula powder, and that translates to 0.15 milligram per kilogram in a liquid formula, but then 2.5 milligrams per kilogram in other foods for adults. The lower level in infant formula accounts for the higher level of exposure and the fact that infant formula can be the only source of nutrition for an infant, so we really have to be more conservative there. One thing to note is that in the supporting documents and in the discussion at the Codex Committee on Chemical Contaminants in Food, the point was made that a level, maximum level in liquid milk needed to be lower than what the level in the finished product infant formula could be. The liquid milk gets concentrated into powders, and as the liquid is concentrated, the melamine itself gets concentrated. So the proposed level for liquid milk was 0.1 milligrams per kilogram. That's not officially adopted into the Codex documents, but it is a good rule of thumb as you're trying to look at the potential for melamine throughout the supply chain in milk and milk powder. There are several countries that have a zero tolerance, especially in the Southeast Asia region, in response to the melamine, to the China crisis in 2008. Many of the countries there set zero tolerance. They set it at the limit of quantitation for the methods, which was 50 ppb, 0.05 milligrams per kilogram. So much lower than what the Codex limits are. But they made the point that it was added as an adulterant and shouldn't be there at all. So they took a zero tolerance standpoint. So it's something to keep in mind as if you're looking at a global supply chain. Regulations around the world don't all align to Codex. Analytically, this is an AOAC meeting, so we can't ignore the analytical aspects of this. From an analytical chemistry standpoint, melamine really changed the way that we look at food and food safety when we're talking about adulteration. Prior to the melamine incident, most methods were based on targeted analysis. We had compounds that are regulated, compounds that we knew we did not want to be in foods, so you develop a very specific method for that compound in the food matrix, and you can do the testing. You can get very reliable identification, good quantitation, usually very low detection limits in a targeted method. The problem is, if somebody is doing food adulteration with something that you aren't aware of, then you'll never see it. So the mass spec methods that are used, the LCMS or GCMS methods, for melamine, there have been immunoassays developed. So there are rapid targeted methods for melamine. The problem becomes what happens if they find a different compound that serves the same purpose. So we always have to be on the lookout for what might be next. Try to think like the food adulteration people might think and figure out what they might do so that we can stay up front. So there's been a lot of effort on non-targeted methods in the last 10 years. We're looking for things that we might not know are there, but we really need to understand if the food is consistent with what we expect it to be. You do a lot of testing up front to prove that the ingredients in the food is authentic, and then you can do testing to assure that you're maintaining that authenticity over time. These are generally based on universal detection methods, spectroscopic methods, maybe NMR, but they give a good way to monitor the food and your supply chain to make sure that everything is still consistent and safe. Just one example of how things have moved in the last 10 years. Because melamine was a high nitrogen compound, really we looked at other high nitrogen compounds. And when I say we, I mean the analytical community in general. There have been multiple publications of methods similar to this. This is one that was developed in our lab. Chromatogram is only 5 minutes. It's based on UPLC mass spec. It gives very good detection limits, and it allows us to look for both melamine and cyanuric acid, as well as some other known high nitrogen containing compounds that may end up as food adulterants in the future. So it's just a way to try to predict what might happen. Somebody could always pick a different compound that's not in this targeted method, and we would still miss it, even though we're trying to incorporate more compounds. So that's just a brief overview of the history, why melamine is important, but also why cyanuric acid is important. Because melamine continues to appear periodically in the supply chain, if we see it, we have to make sure there's cyanuric acid. Melamine as an adulterant, we need to limit and try to get rid of. We don't want the adulterants to be present. But if cyanuric acid is also present, it creates a significant safety issue that we have to immediately take action on to make sure that we're protecting the public. So with that, I'll close. I appreciate, again, the opportunity to speak. If there are any questions, please, you can submit by email. I'm sure that they can forward the questions to me. I'll be happy to respond. And I am sorry I couldn't be there in person. Hopefully in the next year, things will get better and I'll be able to travel and I'll be able to meet you then. Thank you very much.